All right, home stretch. So we talked about the cell cycle, we talked about parts of cell division, and now we're going to talk about a little bit about how this whole process is controlled. Okay, the quick answer is chemicals, right? Chemical reactions control everything inside our body. However, we'll be a little more specific when it comes to the cell cycle. So we need some checks and balances to make sure that the cell cycle continues correctly and in the right procedure, and that it doesn't move on until the previous stage is completed successfully. And so there's some checkpoints, and there's actually lots and lots of checkpoints, and we're very confident that we don't even understand all the checkpoints that happens in this fairly straightforward process that has happened and is happening in your body hundreds and thousands and millions of times. So we're going to talk about three. We've got, and they're all parts that you've already learned about in the cell cycle. So there's a checkpoint at G1, there's a checkpoint at G2, and there's a checkpoint within mitosis. Okay, so let's look at each one just a little bit. So the G1 checkpoint is where it's kind of, there's a point of no return, right? That this is where the cell will have to make the decision of that splitting path between G0, where it's not going to divide, it's going to stay in that suspended animation, not going forward, or there's a point of no return, where we're going to go with this, we're committed to dividing. The only way out of that decision, like if the cell meets this checkpoint and then later cannot, is a word I mentioned earlier called apoptosis. And that means that the cell kind of implodes, it dissolves itself in case, well, there's something wrong with the cell. We need to get rid of it. We don't want bad cells to continue to reproduce and divide. G2 happens during the G2 phase of interphase, and that's after DNA replication. That's after the S phase, which is DNA synthesis. And so the G2 checkpoint is literally enzymes going up and down the DNA to make sure everything's been copied correctly. And it's kind of cool because if there is some damage to DNA, some mutations, or there's been an error in replication, there's actually ways to fix that, which is pretty cool. Because if you think about how many, well, you don't think about how many DNA bases there are, but there's a lot. And the chances of some kind of copying error is pretty high, but yet we fix them as we go. Some of them do slide through, but most of them get repaired. And then we have a mitotic stage checkpoint, and this happens between metaphase and anaphase. And this is that we need to make sure that all of the chromosomes get attached to those spindle fibers so that they can be separated out. If they don't pass those checkpoints, the cell, again, gets a signal to dissolve because of those lysosomes inside. So again, this should be familiar, but now we're going to put in some checkpoints, 3 of many. So here's one in G1, and if it doesn't pass this checkpoint, then the cell dies. Okay, and then there's another one after G2, and if the DNA has not been replicated, or maybe we missed a whole chromosome, it won't go forward. It'll stay here, or it'll die. And then in mitosis between metaphase and anaphase, we're going to check that all the chromosomes have been attached to the spindle. So what turns on and off all of this stuff to start this, to start moving from one phase to the next, we need signals. Now these signals can be any kind of molecule that stimulates an event or inhibits an event. Okay, we usually think of a signal that just stimulates something, but it can also inhibit. It can also prevent something from happening. So of course we have 2 types of main signals. External, which means they come from outside the cell. One example would be contact inhibition, where for the most part cells will stop dividing when they come in contact with another cell. So if you imagine that paper cut on your finger, the cut needs to be repaired, but we only want the cells to continue to divide until the wound is healed. Once one healthy cell touches another healthy cell, we need it to stop. If not, you would have all these folds of extra skin, right? Internal signals can also stimulate or inhibit cell division. And a class of internal signals are called cyclins. And they're the ones that kind of make sure that we go through the cell cycle, like starting up DNA replication. I've mentioned this word, apoptosis, and that means programmed cell death, which is pretty cool. So the cell, ruptures, and in humans, the white blood cells will kind of gobble up all the cell fragments, and this signal can be made from internal or external sources. So there's some apoptosis that occurs because it's very normal in growth and development, like when tadpoles lose their tail and they're a frog. Apoptosis on that line between occurs so that the tail falls off. But also early in human development, fetal development, of course, before you're born, there's actually a thin layer of webbing that goes between each of our fingers. Okay, and then later in development, the line of cells between each finger is killed. It goes through apoptosis so that our fingers are liberated. Of course, there are some humans, and you might know some, maybe you are one, that signal stops a little prematurely, and especially in the feet, they have webs. They have webbing between their toes, sometimes between their fingers, though I will say even before they go home from the hospital, those first few days, the doctor will go ahead and kind of snip them back a little bit to get rid of that. It's a genetic thing usually, so it runs in families. So what happens when the cell cycle doesn't pay attention to these signals? Well, literally, that's the definition of cancer. I know cancer is a big, evil word, and I agree, but scientifically, it's a pretty simple definition. It's a disease where the cell cycle is not working right. And it just keeps going through division after division after division. Now, the cause of that disruption of the normal cell cycle is many faceted. There are many causes to that, so that's the part that gets really complex, right? But all cancer, no matter where it is in your body, is literally the cells can't stop dividing. Sometimes it's because there's a lack of certain signals. Sometimes there's too many signals, okay? So we'll go through a few things about cancer in this part. So if you think about it, as far as we know, cancer starts with one cell. One cell that has one mutation. However, that's not going to cause cancer. That one cell with one mutation is not going to be enough. There's enough checks and balances in our cells that we would suppress that cell. It would not form a tumor. The problem is when that cell that has one mutation changes a little more, and now all of a sudden there's a cell that has two mutations, as we can see here, this little green one. The red ones just have one mutation, no big deal. And then we have this one that has two mutations, and it all of a sudden divides a little faster. And then out of that mass, we have this purple one that has three mutations. And they're not just any mutations, my friends. They're mutations in genes that should be controlling the cell cycle. I want to be sure of that, right? And then that little purple one with three mutations to certain genes all of a sudden become a mass. And those cancer cells, if they are close to the bloodstream, they can get into that bloodstream and move around. If they're close to the lymphatic system, which I know doesn't get much talk about, but it kind of runs parallel with the circulatory system. If it gets into our lymphatic system, then that's how cancer can spread, because that's kind of the superhighway of our body. And I know many of us, probably all of us, have been touched by cancer in some personal way. And so if this is bringing up awful thoughts and memories, you might take a pause, take a deep breath. I think it's important to understand the science behind it, but please note that I'm not trying to be calloused towards the personal side of cancer, because I know there's many, many stories. So these mutations that cause cancer are usually in one of two classes of genes. And I'm oversimplifying this, but I think it's important to kind of dive into this. So there's two main classes of genes that when they get mutations, you're at a much higher risk for cancer. Now, how you get these mutations, it may be a combination of genetics, where you've inherited it since the beginning of your life, But a lot of them come from the environment, that over time, these mutations occur randomly or because of certain chemicals that we are exposed to. One class of such genes are called the proto-oncogenes. So in general, proto-oncogenes are good. They promote the cell cycle, they inhibit apoptosis, and they're supposed to be there, right? However, when those proto-oncogenes mutate, we call them oncogenes. Proto means before. Okay, so we discovered oncogenes, and then we thought, well, yeah, before the mutation, they're good. After the mutation, they can be bad. So oncogenes are one class of cancer-causing genes. And I want you to think about proto-oncogenes kind of being like a gas pedal for the cell cycle. And when your gas pedal gets stuck, that's like an oncogene where the cell can't stop dividing. The other class of genes are called tumor suppressor genes. Again, tumor suppressor genes are good. We have to have them because they're the brakes in the car. They say, hey, stop dividing. We have enough cells. We don't need any more. They inhibit the cell cycle. They promote apoptosis. They're the opposite of proto-oncogenes. The problem is when there's mutations in tumor suppressor genes, now you have a broken brake pedal. And the cell will keep dividing, keep dividing, keep dividing. Do you see the outcome of both of these mutations, whether it's an oncogene or a mutated tumor suppressor gene? We can't stop dividing the cells. So here's a little picture of the receptor. Maybe it's no good, and so it just keeps sending a signal like keep dividing, keep dividing, keep dividing. And then if we have a mutation in a tumor suppressor gene that should be saying, whoa, 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 we got to stop, man. And it doesn't. That's why it's got to be more than just one mutation. It's going to be the accumulation of a few mutations over time. The greatest risk factor of cancer is age, my friends. Just over time, we accumulate those mutations. So some characteristics of cancer cells. They don't go through apoptosis. They stay around forever, or a long time. They don't die like they should. They can form tumors, and those tumors don't respond to stop signs. And they can spread. That's called metastasis. Okay? Angiogenesis means that they can actually trigger the formation of new blood vessels. And when they get new blood vessels, then they get more oxygen, more glucose, and so they can divide even faster. I bet you've heard of these words, benign and malignant. Benign means, well, it is a tumor, but it's contained. It's not going to spread anywhere, but malignant means it's dividing and it's spreading. I'm going to have to speed it up a little bit. I'm talking too long. So cancer treatments kind of go in a few ways at this point. We try to kill the cells or remove the cells that are cancerous that won't stop dividing. Okay, you may have to pause here and read up on this, but these are probably familiar to you. But of course, we're working on new ones all the time. There's two major ideas behind cancer prevention. The behaviors that you might need to avoid, like smoking. 30% of all cancer deaths can be related to smoking. And we're not just talking cigarettes, my friends. Please be careful about the e-cigarettes, vaping, Juul. We don't know anything about that, my friends, and you're putting those chemicals directly into your lungs, directly into your bloodstream. Sun exposure, right? One of the deadliest cancers is melanoma, is skin cancer. And of course, drinking a lot. Okay, that is prone to a few different kinds of cancer. There's also things that we should do that are good, that are promoting good behaviors, like keeping a healthy weight, eating the right kinds of foods, making sure we have the vitamins we need. And so, yeah, and eat cabbage, it says. All right, so please take a moment and review back anything you need, and I will see you next week. Bye.